Hey guys, this is Dr. Boney. This is just a little review of the section of Principles of Genetic Variation in Chapter 4, the Functional Genetic Variation and Protein Polymorphisms. Boy, try to say that 5 times fast. Um, so basically here, again, this is general principles of all the variation that exists outside of the DNA. So obviously, we have DNA, and as you know, some of the DNA codes for proteins and some of it doesn't. But even if the code is the same, sometimes the subsequent proteins that are constructed are not all in exactly the same form, and there's reasons for that. So this is kind of a little bit of a tackling of that particular aspect. So we're going to talk about the origins of where this variation exists. We all do have some variation, and some of that variation in key spots does create polymorphic proteins, or proteins that exist in several different versions. And there's a lot of those. So also DNA repair mechanisms, how DNA does that. That's a pretty involved scenario of many different methods. The scale of human genetic variation, whether it's a particular protein or how it might even affect an entire trait or organ system, etc. And then the consequences of the variation. And so that's just a little bit of the um, starting outline. So let's get to it. This particular image is a good one. And I'm going to use the first example here, the particular database on SNPs, pronounced SNPs. These are single nucleotide polymorphisms. So this particular study, if you go to it, and I'm going to kind of show it to you in a minute, gives us an entire library of SNPs within the human genome. And basically, SNPs are the most common type of variation that are found in people. And at least 1% of the population has to have the same variation in the same location to be considered a SNP versus a mutation. So they happen fairly often, every 300-400 nucleotides, something like that. So there's an awful lot of SNPs that we have. And since 99% of the genome of people is identical, these SNPs are used to provide us a way to study, for example, like the genetic ancestry of differences that are apparent across human races, for lack of a better word. Almost all of these, it's important, have almost no effect on your health at all. They're just ways that we can kind of tell differences amongst people when so much of our genetics is the same. So let's take a look at this first database and see what it looks like. So I just opened up NCBI, and let's pick on gene, and let's just pick maybe BRCA2. You probably have heard of that. That's breast cancer gene. It's a gene to help us to fix errors in DNA replication. So let's hit search and see what we get. And obviously the first one is human, so let's hit on BRCA2. And here is a whole bunch of information about where it is. Chromosome 13, arm Q13.1, number of exons, the whole deal, all kinds of stuff. That's hopefully you've learned some of this. If not, you will in later classes. But if we go down here, this is SNP gene view. We're going to see all the different SNPs that exist. And as you can see here, there are a lot. Ready? Yeah, there's a lot of them. So basically what these are is this tells you exactly where the mRNA position is, what kind of mutation it would be, what the protein residue is, where that mutation occurs, because obviously this was a missense, so it's going to cause a different amino acid change, and there's a lot more information about it. Now, a lot of these do not cause disease, so it's not like, oh my gosh. Most of these have little to no effect, nor are they very, very common. Some of them do, and so some of those inherited SNPs can cause disease, and that's one of the ways that we can diagnose them is through SNPs. So I just wanted to kind of show you one of them to kind of give you an idea about how we can find some of these different SNPs through one of the example databases I showed. So I kind of just touched on this point as well, that obviously some of these are going to have a neutral effect. You might have a missense mutation that might substitute one amino acid for a different one, but if those amino acids have the same relative characteristics of charge and hydrophobicity, etc., then it may have little to no effect on the protein. There may be some that cause silent mutations, so you're going to be producing the same amino acid. Again, some can be harmful and some can actually help depending on what the scenario is for the particular SNP in that case. So this is just a kind of an example here of a relative cladistic relationship, a phylogenetic tree of the FOXP2 protein and where the variation exists. And you can see here that modern human has a lot, basically the same substitutions as Neanderthal. And predictably, as we get farther and farther away, we see that there's different mutations as well. You know, a lot of these are inherited clearly, and at some point either just kind of rode along from chromosome to chromosome, kind of hiding because whatever that particular SNP was had no consequence in the strength or viability of the organisms, or it helped in some particular case. And by the way, remember, not all SNPs are bad, even if they may be bad for a person. Some may be good for some degree of selection environmentally. For example, sickle cell heterozygotes are protected against malaria. If you were to live in sub-Saharan Africa, that would probably be a good thing if you didn't have proper medical care or be able to avoid mosquitoes, that you would have a better chance of not getting malaria, for example. So this is a really good kind of other view of variance. And of course, ultraviolet radiation, as you can see, obviously has a degree of variation. If we have, for example, low exposure to it, some particular versions of genes like this one, which SLC 24A5, has a lot to do with pigmentation. So if you are in an area where there is low UV exposure, you would want to be able to transmit, as it were, components of photons in order to be able to make vitamin D. And so if there is a lot less UV exposure, there is an environmental difference, basically, in not necessarily the gene itself, but fundamental amounts of gene expression that are based on low UV to allow vitamin D3 to be produced in cases of lower UV. Sorry. Again, there is, the one I really like is the second one. I'm not going to go through all these, but Tibetan populations, really interesting, have a gene involved in hypoxia response. And hypoxia obviously is not enough oxygen because they are in very high altitude areas. They basically have a very high density of blood capillaries, which would make sense, and provide against hypoxia. So all of these are environmental things that over time gave some degree of benefit to some people in the population versus others, and therefore a higher chance of procreating and passing on that particular change. And so this is just a couple examples of one of them. Last one I do want to talk about really quick, obviously, is intake of fresh milk. It is not a natural thing for adults to be able to drink milk. Milk, as you know, in mammals exists to feed babies, either human, cat, dog, doesn't matter. But some people have been able to maintain the ability of lactase production beyond having been a younger person. And so there's the LCT gene that has a lot to do with that. So pretty neat stuff. Again, just another particular example, not going to go into huge detail here, but you can see that gene copy number also has a lot to do with, for example, alpha amylase and high starch diets. You can see that there's a difference in the number of copies of the gene, which would of course have a difference in the phenotype. This is actually a little bit different. The picture may look a little overwhelming, but you can definitely read part of your book about what this is. But as simple as I can make it, it's when a new mutation happens to be good for the organism. increases in frequency, and we can see the mutation happening basically on line 3. This is one of the variants out of the 1, 2, 3, 6 variants here. And basically what happens is this thing becomes permanent and leads to a reduction of other variations of nucleotide sequences around that particular individual diamond mutation where it says advantageous DNA variant. So what that really means is that some of these other genes that are linked, and of course linked mean on the same chromosome, can also be passed on as well, and we call it basically hitchhiking. So they can become fixed. And so you can see here that this third variant was just one of 3, but for whatever, this particular mutation became an advantage because it was a positive mutation, beneficial, and now we can see after time there is still a little bit of variation left here, especially in the outer exon areas, or other genes, I should say. But we can see it's now fixed because it was of benefit. And so that's what a selective sweep variant is. Another way that we can depict genetic variations, this is for SLC 4A5, which is a member of the sodium bicarbonate co-transporter family. Boy, that sounds exciting. And this basically, obviously if it's a bicarb, you can figure it's got something to do with pH regulation. It's how the cells can regulate their pH. They need to do that as well. It's a transport protein. So you can see here that even amongst different, I should say, genetic families or ethnicities, we see that there are major differences in where these variants occur. Whether they're caused by an environmental situation or not, I mean, obviously there would be more study to that, but you can see the level of heterozygosity that's involved in this particular gene and its variants. So another diversity of protein, one can be caused by gene duplication. Now, gene duplication has a lot of evolutionary implications, but to keep it as simple as possible, and you can go back in the book for more detail, obviously, but it's basically how what a region of DNA that codes for a gene is basically copied. So there's an area of the chromosome that's photocopied, and then that photocopy gets put into the chromosome also. So for example, if there was a particular gene or set of genes, usually a gene, there would be a copy, and then there would be now 2 copies of that gene on the same chromosome. And that happens typically because of some sort of recombination, error, obviously error in recombination. But these duplicated genes have immunity to selective pressure, basically because there's a functioning gene that's working. In other words, the duplicated gene a lot of times will have mutations. And normally, just let's back the truck up a little bit, hopefully you do realize that coded genes tend to diversify way less than non-coded areas of DNA. And the reason why, if there's a structural function to a particular gene, if you have a mutation and then that gene produces a protein that doesn't function anymore, that's not an advantage for the organism. So, you know, introns and areas of all kinds of, quote, junk DNA, it's not really junk, that don't code for proteins, non-coding DNA, there tends to be way more diversity. So this duplicated gene can almost act like it gets free rein and is not under the same pressure to not mutate, because there's a version of the gene that's producing the proper protein. So sometimes those random mutations in the copied gene could be of advantage to the organism and could therefore create a different trait or another version, a polymorphic trait. So that's one example. Alternate processing of a single gene could refer to a lot of things. In this case, it could be referred to isoforms, basically skipping of exons. If a particular gene has like 6 exons, like 1, 2, 3, 4, 5, 6, there might be versions that make 1, 2, 3, 4, 5, and 6. There might be one that's 1, 3, 4, 5, 6. There might be one that's 1, 4, 5, 5, 6. So these are things that can cause protein diversity fundamentally. So this is a picture of, again, variation of sets of genes in relationship to olfactory receptors, and you can see here that there are different kinds of particular SNPs that exist, and also here you can see CNV is copy number variation. So you can see, for example, in 11G2, The most common variation happens to be homozygous for a particular deletion in a copy number. Whereas on the other side of the spectrum, the intact version, the homozygous for 2v2, that particular gene doesn't have a lot of variation in it. So finally, post-transcriptional induced variation, boy, that's a big fancy word. This goes back to the alternative splicing I was talking about. For example, like in isoforms, isoforms are basically versions of genes that have, actually really versions of genes that become versions of proteins, many of which have a functional property, some of which don't. There are some diseases that are caused by isoforms of a particular functional gene, but these are RNA or later. So even though the DNA is the same, the subsequent protein is going to be different. Exon skipping, again, is one particular way of getting an isoform by incorrect splicing, and also alteration sometimes by the editing of RNA. So these can all cause variation as well. So hopefully you enjoyed this, and I will talk to you later.